Hello and welcome to Journal Club today. So today we've got Haider Zahur who's going to talk to us about the ALBIOS trial. So I'll hand you over to him to start on the trial.
Yeah, thank you, Sahim. So I'm Haider. I'm one of the dual ICM distributed trainees. Today I'm presenting on this ALBIOS trial that was published in New England Journal of Medicine in 2014, which actually looked into the albumin replacement in patients with severe sepsis or septic shock. Just before going into the ALBIOS itself, I thought we just run through a bit of a background of the need for this this trial. So because it will be around the albumin, so let's just have a bit of talk about the albumin itself, just a brief one. The human albumin is the main protein in the human plasma blood, which roughly makes about 50 to 70% of the human plasma protein. And it carries different functions, but the most important one that is that we are going to talk about today is it binds into many endogenous and exogenous compounds, including the fatty acids and the metalines and pharmaceuticals, and it actually makes a role in the oncotic pressure. It actually does regulate a bit of an oncotic pressure. That is why there has been discussion for long about the role of albumin itself in patients with severe sepsis and septic shock, which makes it more important. We already know that for decades we have been giving albumin administered to the patients with the hope that it might make any significant difference to their blood pressure, especially in the patients who are in sepsis. And this trials of albumin goes way back in late 90s, and the initial trial was to look into the role of albumin in critically ill patients, which was a randomized controlled trial which was published in BMJ in 1998. This trial showed at that point that there was no evidence that the albumin administration reduces the mortality in patients with clinically ill patients with hypovolemia burns. However, it does mention that the human albumin in critically ill patients has got worse prognosis outcomes. And then they put up this comment as this should be urgently reviewed and that it should not be used rigorously in patients, in ITU patients, which was again the concept that has been commonly practiced back in early 2000. Follow that, there was then, follow this 1998 BMJ publication, there come a major study, which is the SAFE study, which was again a multi-center randomized double-blind control trial, which actually has studied the effect of fluid resuscitation comparing albumin to that of saline and crystalloids in ICU patients. And the summary of the SAFE study is they did not see significant difference in the 28-day mortality between the two subgroups, the albumin group and the normal group, the control group, which were actually treated by the crystalloids, which were again contradictory to what we have seen initially in the BMJ publication in 1998. Following that, there was another publication by University of Brussels, which was then later on published in Critical Care Medical Journal. And they did, previously studies, they did not set up a standard for the albumin. They just thought albumin is effective. In this study, they actually have set a standard, and the standard they find out that there might be a potential benefit in patients who we treat with albumin during an ITU with a sepsis if we maintain a level of albumin of more than 30 gram per deciliter in these patients. And they have actually found that this seems to have proven some benefit compared to what the previous study did show. Now, because we have seen that there's been two contradictions, so one in 1998, they say it is actually harmful, but the SAFE study in 2004 said it is not harmful, but it's not over convincingly helpful. But then this Critical Care Journal publication mentioned that actually it can be helpful if you maintain a protein standard protein level. So what they did in the ALBIOS study is they actually looked at the clinical question they looked at to it was whether they can give 20% albumin and it maintains any benefit when compared to crystalloids when they maintain a serum albumin of more than 3 grams per deciliter, and then they calculated the mortality in 28 days. Again, this was a multi-center randomized controlled clinical trial, and then they stratified to the participant in ICU in the time of onset and the randomization, which should be something between 6 hours and less than 6 hours participating compared to more than 6 hours. But the only thing was this was an open label non-blinded study, and it was upon the discretion of the consultants participating in those trials is to put either into the albumin group or to put into the control group. So it was carried out in 100 intensive care units in Italy, and the study was conducted between August 2008 to about February 2012, with a total participants of about 1,818. They developed a inclusion and exclusion criteria for the people who was part of this trial, and any adult who met the clinical criteria of severe sepsis, which is systemic inflammatory response syndrome, plus proven or suspected infection with at least one organ dysfunction, as measured by the SOFA score. And the exclusion criteria was head injury, patients with congestive heart failure, or a patient who has been over 24 hours since the inclusion of the criteria was a part of this study, and also someone who's got objection to blood products, like we know the Jehovah's Witness, or people who require albumin for their specific clinical reasons. People with acute liver disease, sometimes they need as a part of their the treatment. So those are the people who were excluded out of it. So then they divide into 2 groups, the intervention group. So the intervention group received both the 20% albumin crystalloid, while the control group only received the crystalloid solution. So what they did is they gave about 300 ml of 20% albumin daily to maintain the serum albumin of 30 gram per liter or more. And we have already discussed that the standard of 30 gram per liter has come from a study that was done in Brussels. And then they also give crystalloids to these patients as well, but it was upon the physician discretion rather than anything else to decide whether they need any further fluids beyond the 20% albumin. And the 20% albumin was actually calculated about 4.3% of the median daily average that the patient received during his course of treatment, and the median of the total fluid for the first 7 days that the patient received was about 3,700, and about 4.3% of that was 20% albumin. This compared to the control group, where only the crystalloids were given. Again, this was left to the physician discretion, but when they mentioned the median total daily fluid was about 825 mils, which was not significantly different to that of the intervention group in the first 7 days of the treatment. So they calculated the mortality in the first 28 days, and they find that there was about 31.8% mortality in the albumin group versus the 32% in the crystalloid group. So actually the mortality at the 28 difference between the 2 groups was not significant. And the secondary outcome, they calculated the mortality at about 90 days, which is about 41.1% in albumin group versus 43.6% in the crystalloid group. This study has got, so this is a pragmatic study directed relevant to the clinical practice. And the reason why I said that is when in the start of my presentation, I gave you a reference of 3 previous studies. One was the 1998 PMJ study that mentioned it was actually harmful to give albumin to these patients. Then the SAFE study says, well, it's not actually harmful, but it's not convincingly helpful. And then the Brussels study actually said that it is helpful only if you maintain a serum level of 3 gram per liter. And the other thing was, out of those 1,818 patients, only the 29 participants, which it makes about 16.6% of the total, well, actually well below the primary outcome rate. But it was, you can argue that in the SAFE study, they did mention that there wasn't any significant difference, but they still went on to conduct this large study for 4 years in 100 different ITUs. And the other thing was, it was a non-blinded study, so it was upon the physician discretion of to place which one in the control group and which one in the albumin group, which could have biased towards either the intervention or the control group. But at the end, it didn't make any significant difference in terms of the treatment. The conclusion of the study was, so the use of 20% albumin in patients with severe sepsis will improve the hemodynamic indices. We already know that the serum albumin maintains about 50 to 70% of the total human body protein, and they play a major role in regulating the oncotic pressure. But however, they do not seem to be effective in terms of reducing the mortality in patients with severe sepsis. They also find that it is safe, but not recommended the use of routine fluid resuscitation with albumin in patients with severe sepsis. And the target of serum albumin of 30 gram per liter or more does not appear to have made any survival advantage both in 90 days, in 28 days, and in 90 days from the ALBIOS study, which was conducted. Yeah, that was it. It was a small study, but I thought to make it more interesting to add a bit of the previous studies and the albumin, just to give you a rough idea of where the standards came from and why was the need of conducting this study. I'm happy to take any questions.
Thank you very much for your presentation. Very good presentation, very interesting. I'll just open up to the floor. I think Johnny had quite a few comments, which you can see in the chat. We've got a few posts on the Critical Care Northampton website, and there's also a link to the blog post as well there. Does anyone have any questions for Haider?
I'll come in if it's all right, Sayam. So, Haider, do you think it's practicably easy to maintain albumin levels of 30 in critical care patients who are septic?
Well, I think so. All of them have been in their booths. Their albumin has always been in their booths, and they must have given a lot of albumin really to maintain this 30 gram per liter. I've hardly seen any patient maintaining at that level here in ITU. So I think so they must have put a great deal of effort to bring it up to that level.
Yeah, because that's my thought. I think that, you know, the averages of 3,800 and 3,700 consecutively of total fluid input per week, they mustn't have given anything else whatsoever. They must have had to rationalize drugs and all kinds of things in order to get that target. So it's a pretty big achievement, I would say, that's not practicable.
Yeah, I mean, when I read the level, and I was like, "Ooh, well, that might be means," and especially if you're taking about one organ that needs support. So yeah, I thought it was a very convincing high level, and that's probably why they invested 4 years and 100 ITU centers into it, that they can practically go into any benefit, which unfortunately they couldn't get any.
And I've got one more question, if that's all right. So the cost implications of providing this level of albumin therapy mustn't be small either. Have you got any comments on that?
They didn't mention much about the costing, and I think that was the other thing that was probably lacking in the paper. So they didn't mention about the cost of what it would have done with someone maintaining this much high amount of albumin. I couldn't read it through when I was doing the paper study. But I presume it will be high. I presume it will be very expensive doing so for this large number of patients.
Okay, have we got any more questions?
Italian.
Any more questions from anyone? Doesn't look like it. Thank you, Haida, for your talk. Very good talk. And next week we will have another journal club. Okay. Does anyone have any closing comments or anything like that? Okay. Thank you.